Assalamu alaikum students, how are you? The next topic we are going to discuss is the multiple alleles. First of all, what the alleles are: the alternative forms of a gene that occupies the same chromosome locus but showing contrasting characters are called the alleles. A gene mutation may produce many different alleles of a gene. Some genes may have as many as 300 alleles. All such alternative forms of a gene whose number is more than 2 are called the multiple alleles. We all know and we know about the genes. Genes consist of 2 alleles, but when some mutation occur, then 3 forms of a gene are produced, and these are called multiple alleles. Any 2 of these multiple alleles can be present in the genome of a diploid organism, but a haploid organism or a gamete can have just one of them in its genome. And gene mutation produce many different alleles of a gene, and some genes also have as many as 300 alleles. And all such alternative forms of the gene are called the multiple alleles. Next one is the ABO, the first discovered multiple allelic blood group system in man. ABO blood group system was discovered by Carl Landsteiner in 1901. ABO system has 4 different phenotypes which are distinct from each other on the basis of specific antigen on the surface of the RBC. A person having antigen A has blood group A. A person having antigen B has blood group B. A person having both the antigens A and B has blood group AB. But a person having neither antigen A nor B would have blood group O. There are 4 types of blood group: blood group A, blood group B, blood group AB, and blood group O. Person containing antigen A has blood group A. Person containing antigen B has blood group B. Person having both antigen A and B has blood group AB, but the person neither containing any antigen, either A or either B, would have the blood group O. These are called multiple alleles. Any 2 of these multiple alleles can be present in the genome of a diploid organism, but a haploid organism or a gamete can have just one of them in its genome. And gene mutation produce many different alleles of a gene, and some genes also have as many as 300 alleles. And all such alternative forms of the gene are called the multiple alleles. Next one is the ABO, the first discovered multiple allelic blood group system in man. ABO blood group system was discovered by Carl Landsteiner in 1901. ABO system has 4 different phenotypes which are distinct from each other on the basis of specific antigen on the surface of the RBC. A person having antigen A has blood group A. A person having antigen B has blood group B. A person having both the antigens A and B has blood group AB. But a person having neither antigen A nor B would have blood group O. There are 4 types of blood group: blood group A, blood group B, blood group AB, and blood group O. Person containing antigen A has blood group A. Person containing antigen B has blood group B. Person having both antigen A and B has blood group AB, but the person neither containing any antigen, either A or either B. But a person having neither antigen A nor B would have blood groups O. ABO blood system has 4 different phenotypes. These groups are different from each other on the basis of antigen. Antigens are present on the surface of RBCs. Bernstein explained the genetic basis of ABO system in 1925. This blood group system is encoded by a single polymorphic gene I. On the chromosome number 9, it has 3 multiple alleles: IA, IB, and I. What is the meaning of polymorphic? Polymorphic with many forms. So gene I has many forms and it is present on the chromosome number 9. gene I undergoes mutation and 3 mutation, and as a result of 3 mutation 1 produces IA, mutation 2 produces IB, and mutation 3 produces small I. Alleles IA specifies production of antigen A. Same case, allele IB specifies the production of antigen B, but allele I does not specify any antigen, and their dominance relations are interesting too. Alleles IA and IB are co-dominant to each other because each express equally in IA IB heterozygous to produce AB phenotypes. But allele I is recessive to both IA and IB. Therefore, IA or IA small I genotypes will produce phenotypes A. Similarly, IB or IBI produces phenotypes B. The homozygous small I will produces the phenotypes O. इस पैराग्राफ में हमने ये डिस्कस करना है कि आई ए जो है वो एंटीजन ए को स्पेसिफाई करता है, उसकी प्रोडक्शन करता है। आई बी जो है वो एंटीजन बी की, और स्मॉल आई जो अलील है वो किसी भी एंटीजन को स्पेसिफाई नहीं करता। और इनके आई ए और आई बी दोनों आपस में को डोमिनेंट हैं और एक ही कंडीशन में एक्सप्रेस दोनों अपने आप को एक्सप्रेस करते हैं इन द फॉर्म ऑफ ए बी ब्लड ग्रुप। लेकिन जो स्मॉल आई है दिस इज़ रिसेसिव टू बोथ आई ए एंड आई बी। देयरफॉर जो है ये जीनोटाइप्स जो होती हैं, अगर कैपिटल ए आई के साथ है यानी होमोजाइगस कंडीशन में ए मौजूद है और हेटेरोजाइगस कंडीशन में आई ए स्मॉल आई के साथ मौजूद है, देन क्या होगा, फिनोटाइप ए प्रोड्यूस होगी। इसी तरह जो है अगर बी जो है होमोजाइगस कंडीशन में मौजूद है और हेटेरोजाइगस कंडीशन में स्मॉल आई के साथ मौजूद है, देन उसकी फिनोटाइप जो है वो बी होगी। सेम इज़ द केस कि अगर हम होमोजाइगस कंडीशन में स्मॉल आई इकट्ठे हो गए हैं स्मॉल आई की फॉर्म में, देन फिनोटाइप्स ओ जो है प्रोड्यूस हो जाएगी। द ब्लड ग्रुप alleles start their expression at very early embryonic stage and keep on expressing themselves till death. Therefore, the blood group phenotype of a person never changes throughout the life. Next, the antibodies of blood groups. Blood groups anti-A and anti-B antibodies appear in the plasma during the first few months after the birth. They are naturally occurring in the absence of corresponding antigens. The blood serum of A phenotype contain anti-B antibodies. They will agglutinate or clump any RBC which have B antigen on them. B phenotype contain anti-A antibodies in the serum and agglutinate any RBC with antigen A. Phenotypes AB has neither anti-A nor anti-B antibodies in the serum. The serum of O blood type contains both anti-A and anti-B antibodies. The blood serum containing antibodies is called the antiserum. And what is the serum? Serum is the light yellow fluid that separates after clotting of. Some further explanation of ABO blood type. ABO blood type in the human is determined by 3 alleles IA, IB and I. IA and IB are co-dominant alleles. Both IA and IB are dominant to allele I. And the genotype and the blood type IA and IA, if A is present in the homozygous condition, then blood type will be A. If they are present in the heterozygous condition IA and I small I, then it will be A. If B is present in homozygous condition, then the blood type will be B. If B is present in heterozygous condition with small I, then blood type will be B. Same is the case if IA and IB are collectively present in a person, then the blood type will be AB. And if small I and small I are present in homozygous condition, then blood phenotype will be O. Students, now focus on this table. If the person has blood group type A, then genotypes will be either A in homozygous condition or capital A in heterozygous condition. An antigen on the RBC will be A, or phenotypes either A homozygous or either A heterozygous. And the antibody in the plasma will be B. In both forms, either in the homozygous form, either in the heterozygous form, the antibody in the plasma will be B. Same case, if the person has blood group B, then the genotypes will be either B in homozygous condition, either B in heterozygous condition, and the antigen in both forms, either in homozygous form, either in heterozygous form, will be B. Same is the case, antibody will be A in both homozygous and heterozygous condition. And blood group AB genotypes will be IA and IB in collective form, and antigen on RBC will be AB, and phenotypes will be AB and antibodies none. If the person has blood group type O, then the genotypes will be small I in homozygous condition. Only in homozygous condition II is present. No antigen on the RBC, and phenotypes will be O, and antibody in the plasma will be AB. This is the explanation of blood groups and their genotypes and antigens, phenotypes, and antibodies in the plasma. Now the transfusion of the blood. Any blood transfusion is ideally safe if it does not cause agglutination in the recipient. Agglutination leads to the serious result because clumped cells cannot pass through the fine capillaries. The blood samples of the donor and the recipient are cross-matched for compatibility before giving transfusions. If incompatible blood is transfused, dangerous hemolytic reaction occur. Either the antibodies of the recipient destroy the RBC of a donor, or the antibodies of the donor hemolyze the RBC of the recipient. Blood group A can be transfused only into the A and AB recipient because they do not have anti-A antibodies. Blood group B can be transfused only into the B and AB recipient as they do not have anti-B antibodies. AB blood can be transfused only into the AB recipient because they have neither anti-A, neither anti-B antibodies nor anti-B antibodies. O blood group has neither A. O blood has neither A nor B antigens, but it does have anti-A and anti-B antibodies. An O recipient can only be given transfusion from a donor O. Phenotypes O can also be used as a donor for small transfusion to A, B, and AB recipient, but because donors' antibodies are quickly absorbed by other tissues or greatly diluted in the recipient blood serum. O blood group individuals are called universal donors. AB blood group individuals are called universal recipient because they can receive transfusion of the blood from any of the four blood groups. Now, if we summarize the topic, then we can say blood group A can be transfused only into A and AB recipient. Blood group B can be transfused only into B and AB recipient. AB blood can be transfused only into AB recipient. O recipient can be given transfusion from a donor O. So the O blood group individuals are called universal donors, and AB blood group individuals are called the universal recipient because they can receive transfusion of the blood from any of the four blood groups. The yellow box, A and B antigen can also be present in the saliva and other body fluids of some person called the secretors. Secretors have dominant sector gene SE on the chromosome number 9. It means that antigens are present in saliva or other body fluids, and these persons are called the secretors. Now, the significant uses of blood group studies. Genetic analysis on the basis of blood groups helps in solving cases of disputed parentage. It can only be used to prove that an individual is not the parent of a particular child. Child of AB, IA, and B cannot be the child of a parent of phenotypes O. Similarly, a man of B phenotype cannot be the father of a blood type A child whose mother is of phenotype O. His father could either B, either be A or AB phenotypes. Now students, solve this activity. This is all about the blood groups. Now the yellow box, blood type data is not sufficient evidence for disputed paternity. Most modern DNA fingerprinting test is more seriously considered for a legal decision. Now students, for further explanation, पेरेंट ब्लड टाइप्स देख लेते हैं कि पेरेंट्स की अगर ब्लड टाइप्स क्या होंगी तो पॉसिबल चाइल्ड की ब्लड टाइप्स क्या हो सकती हैं और नॉट पॉसिबल चाइल्ड की क्या हो सकती हैं। फॉर एग्जांपल, अगर पेरेंट्स की मदर एंड फादर की ए एंड ए है तो जो उनके चाइल्ड्स होंगे या उनके किड्स होंगे उनकी ए या ओ हो सकती है। सेम इज़ द केस के बी और ए बी बिल्कुल भी नहीं हो सकती। इसी तरह अगर ब्लड टाइप्स ए एंड बी हैं तो उनके पॉसिबल चाइल्ड्स ए बी या ओ हो सकते हैं और पॉसिबल चाइल्ड ये चारों उसी में आ गए अगर ए एंड बी के हैं। एंड थर्ड वन देखें, ए एंड ए बी है अगर पेरेंट्स की ब्लड टाइप्स, देन जो है पॉसिबल चाइल्ड्स की ए बी या ए बी कलेक्टिव फॉर्म में हो सकते हैं। नॉट पॉसिबल चाइल्ड में क्या हो सकता है, उनकी ओ बिल्कुल भी नहीं हो सकते। इसी तरह पेरेंट ब्लड टाइप्स अगर ए एंड ओ हैं देन उनके जो पॉसिबल जीनोटाइप्स हैं बच्चों के तो ए या ओ हो सकते हैं, बी और ए बी तो बिल्कुल नहीं हो सकते। सेम इज़ द केस नेक्स्ट वन देखें, अगर दोनों पेरेंट्स के बी ब्लड टाइप्स हैं देन बी या ओ हो सकते हैं और ए एंड बी बिल्कुल भी नहीं, ए एंड ए बी बिल्कुल नहीं हो सकते। सेम इज़ द केस अगर पेरेंट्स की बी या ए बी हैं तो ए बी या ए बी कलेक्टिव फॉर्म में हो सकते हैं, ओ बिल्कुल नहीं हो सकते। अगर पेरेंट्स की बी या ओ हैं देन बी या ओ हो सकते हैं पॉसिबल चाइल्ड के जीनोटाइप्स उनके ब्लड टाइप्स, लेकिन उनके ए या ए बी बिल्कुल नहीं हो सकते। सेम इज़ द केस अगर दोनों पेरेंट्स के ए बी ब्लड ग्रुप्स हैं देन जो है बच्चों की ए बी या ए बी हो सकते हैं, ओ बिल्कुल नहीं हो सकते उनके ब्लड ग्रुप्स। सेम इज़ द केस नेक्स्ट, अगर पेरेंट्स के ए बी एंड ओ हैं तो उनकी पॉसिबल जो फिनोटाइप्स हैं ब्लड ग्रुप्स की उसकी ए या बी हो सकते हैं, ए बी और ओ नहीं हो सकते। सेम इज़ द केस अगर दोनों पेरेंट्स के ब्लड ग्रुप्स जो हैं ओ हैं देन जो है पॉसिबल जो उनकी ब्लड ग्रुप है वो ओ हो सकता है, ए बी या ए बी जो है वो बिल्कुल नहीं हो सकते।